Hi class, we are going to go into chapter 7, which is the central nervous system. I'm going to switch to screen sharing mode here and get this fired up. There we go. So we're talking about the central nervous system. Here are our objectives. By the end of the week, you should be able to do all of these things. Lots of objectives for this unit. Okay, let's move into the overall organization. First of all, you have the central nervous system, which is just the brain and spinal cord. The job of the brain and spinal cord is to integrate, process, and coordinate all of the sensory information that's coming in and then send out responding motor signals. There's also the peripheral nervous system, which is sometimes called the PNS. This is everything outside of the central nervous system. This includes the sensory neurons and the motor neurons. These have special terms associated with them. The sensory neurons are called the afferent division. The afferent is the arriving information to the brain. It's all arriving. The motor is the efferent division of the central nervous system, of the peripheral nervous system, rather. So let's talk about receptors. We have different receptors in our body. We have somatic sensory receptors. These are touch, position, pressure, pain, and temperature. This is what we typically think of when we think of receptors. Then we have visceral sensory receptors. This is what's going on inside of us. These are our monitoring, our digestive and our respiration, our heart rate, all the things that we're not aware of. Then you have special sensory receptors. These are taste, smell, olfaction, right, balance, hearing, etc. So the motor division of our peripheral nervous system is carrying out motor commands to effectors. Effectors are the target organs. So there's the somatic nervous system. These are our motor neurons going to our skeletal system. This is what we were talking about in the last chapter with the neuromuscular junction and motor units. Then you also have the autonomic nervous system, or ANS. This is all of the information that we're not aware of, all of those motor commands, like our smooth muscle tissue, for instance, our intestines. Yes, ma'am, what do you need?
I want something else to eat.
Okay, I will be there in just a minute. This is also our cardiac tissue, glands, adipose tissue as well, all under autonomic nervous system control. So this is a general overview. You have receptors coming in, bringing information in from the outside world. This is cold, this is hard, this is soft, this is painful, whatever. That's all going to special sensory receptors, traveling through the afferent sensory nerves up into the central nervous system where information processing is going on. This is where your brain says, is this important? Do I really need to know this? Do I need to respond to it, etc.? And then responding efferent commands can either go out through the somatic system to our skeletal muscles where you will, remove your arm from the hot stove, or the autonomic nervous system where you might push food along through your intestines. So let's talk about the actual neurons themselves. The neurons are very specialized. They have dendrites. Dendrites are very short branches from one end of the neuron. These are the receiving end of the neuron. This is where the information is coming into the neuron. Then you have a cell body. This is where the nucleus and most of the organelles are located. Then the axon is carrying the information away from the cell body toward another cell. The axon hillock is the little enlarged portion of the cell body just before the axon. The collaterals are branches off of the axon, and the axon terminal is at the synapse, that special gap between the axon and the whatever cells, the muscle cells or the endocrine cells or the skin cells or whatever. The presynaptic cell is the cell on one side of the synapse, and the postsynaptic cell is on the other side of the synapse. And all materials are transported through the axon through something called a neurotubule. So here you see what I'm talking about. Here is the cell body. These are the dendrites, highly branched, communicating and receiving information either from receptors or from other neurons. On the presynaptic cell, we have this axon going to the postsynaptic cell, our axon terminals. The synapse is the gap in between the presynaptic cell and the postsynaptic cell. Collaterals are the branches off, and the axon hillock is this enlarged area just before the axon. There are some structural variations between neurons. What we just saw here is called a multipolar neuron. That's the most abundant type of neuron in the body, but you also have bipolar neurons, which have two processes on them. These are in special receptors like in our eyes. You also have unipolar neurons. These are these would have a fused dendrite and axon. And the multipolar neurons are like what I just showed you, with 2 or more dendrites and 1 single axon, the most common. So here's the bipolar with a very long dendritic process and the dendrites. Here's a unipolar where the cell body is off to the side and the dendrites and axon are fused. And then the multipolar is the most common, where you have a dendrite all connecting to the cell body, then to the axon terminals. So let's talk about neural stem cells. These do exist in the adult nervous system, but they're very inactive. In our olfactory, in other words, our sense of smell could go away. Those neurons don't replace themselves. The retina of our eye, same thing, not replacing themselves. The hippocampus, which is a memory area of our brain. Most of the central nervous system neurons will not undergo mitosis. They will not be replaced after they have been destroyed or diseased, but the stem cells are there. They're just very inactive. So one thing is, how do we activate them? High area of research. These 3 questions you can answer by pausing the video and going back and reviewing. Let's keep going. Along with the neurons are neuroglia. Neuroglial cells are also called glial cells. They're just the support cells. They support and protect the neurons. This is about half of our nervous system. So half of our nervous system is made of neurons. The other half is made of these special neuroglial cells. There are 4 main types: astrocytes, ependymal cells, microglial cells, and oligodendrocytes. So let's talk about ependymal cells first. Ependymal cells form the lining of the chamber where our brain and spinal cord are found. They produce, they monitor, and they help circulate cerebrospinal fluid, CSF. Microglial cells are related to monocytes and macrophages. These are kind of mobile patrols. They're surveillance cells. They're going out and gobbling up any bacteria, any invaders. They're also removing cellular debris and waste products. Then you have astrocytes. Astrocytes are little star-shaped cells that interlock. They maintain what we call the blood-brain barrier. This is why the nervous tissue in our brain and spinal cord is actually isolated from our blood. It's just the cerebral spinal fluid that surrounds our brain and spinal cord. That barrier is maintained by astrocytes. They provide support for ion and nutrient exchange between the interstitial fluid in the blood and the CSF. They also reabsorb and recycle any neurotransmitters and form scar tissue. Oligodendrocytes produce myelin, and we'll talk about the function of myelin right now. Oligodendrocytes basically wrap around the axon and they form something called a myelin sheath. It's basically like a fatty protective layer. Axons that are myelinated have myelin sheaths. It's basically little sections called internodes that wrap around the axon. Then the gaps in between these wrapped sections are called nodes. Typically, these myelinated axons appear white, or they make up what we call the white matter of our central nervous system. Unmyelinated axons are not completely covered in myelin, and these appear gray. The cell bodies are not covered in myelin. The dendrites are not covered in myelin, so they appear gray. The axons typically will appear white. So we have white matter and gray matter in our central nervous system. And here you see what I mean. Looking at a cross-section of the spinal cord, you can see exactly what I mean. You have this white matter and then the inner gray matter. That gray matter is made up of all of these cell bodies. The white matter is where these myelinated sheaths are. And they're basically special cells that wrap around the axon. And you can see the node is this little spaces that are unmyelinated, and there's a few of those. And then the myelinated spaces are called the internodes, and we'll talk about those functions in a little bit. So you can pause the movie or pause the video and go back and answer these 3 questions. Let's talk about special oligodendrocytes called Schwann cells. Schwann cells are in the peripheral nervous system. They have an outer surface called a neurolemma. A single Schwann cell can form the myelin sheath around one internode for one myelinated axon. So it's just one little patch is one cell, another little patch is another cell. The Schwann cells can also enclose parts of unmyelinated axons to help stabilize the axon. Satellite cells will help surround the cell bodies and the peripheral nervous system, and they basically form little clusters around the cell bodies, and we call those cell clusters ganglia. Here you see what I mean. These are Schwann cells, and they're just wrapped right around that axon, forming little insulated bubbles. You can see those nodes in there, those little gaps in between the myelinated areas. And this is the satellite cell surrounding the cell body. There's a bunch of them together. They are called a ganglia. So let's talk about the functional classifications of neurons. We've talked about the structural neurons, right? Multipolar, bipolar, unipolar. We've talked about the functions of them. We've talked about the types of nerve cells in general. You can have a neuron or a neuroglia. Now we have functional classes. So these nerve cells can either be sensory, interneurons, or motor neurons. So the majority of our cells are interneurons, about 20 billion interneurons compared to 10 million sensory and 500,000 motor. So you don't have to know numbers. It's just to give you a perspective here. Our sensory neurons are unipolar. So they have cell bodies in what's called a sensory ganglia. All the cell bodies are usually clustered right around each other, protected by those special cells. They deliver information through afferent fibers, arriving fibers. They arrive to the brain, right? They are special somatic sensory receptors called external receptors. This is exchanging information about our external environment. Then we also have proprioreceptors telling us about our body position and body movement. This is how I know that I'm sitting in a chair, that my legs are crossed, and I'm talking with my hands, right? It's all through proprioception. Then you also have visceral sensory neurons, which are monitoring internal conditions, like my blood pH, like my blood pressure, like whether or not my breakfast has digested, etc. Interneurons are receiving information from the peripheral and from the central nervous system. So this is basically, these are our strategic neurons. This is the planning. This is the organizing. This is the learning. This is the memory. This is the interconnection between what's coming in and what's going out. It's where what we call information processing happens. So let's talk about motor neurons before we get too much into information processing. Motor neurons carry information from the central nervous system to the outside effectors, to our peripheral effectors. It's called an efferent fiber, or our motor neurons. There are somatic motor neurons, which go to our skeletal muscle. These are under voluntary control. The cell bodies of these are within the central nervous system, and then the fibers are very long and extend all the way through the body. You can imagine how long the fibers have to be to go from your lower vertebrae all the way, or your lower parts of your spinal cord, I should say, all the way down to your toes, right? So these efferent fibers are very, very, very long. There's also visceral motor fibers. These visceral motor fibers go to smooth muscles, they go to glands, they go to your heart, they go to your adipose tissue, and they communicate with something called the autonomic ganglia. And we'll be learning all about these things as we go. So here's where all this information starts. It starts with the receptors. It can travel through our afferent fibers, either through sensory ganglia of our periphery, and depending on if we're talking about autonomic or external, so it could go to different areas. Either way, it's going to end up in an interneuron. Those interneurons will kind of process and sort the information, determine where it needs to go. It can either respond through a visceral motor neuron or through a somatic motor neuron. Either way, it's going straight back down to the effectors, either a skeletal effector or a visceral effector. Notice that some of these internal receptors go right over to visceral neurons. They don't go up to the central nervous system and go through an interneuron. These would be some reflexes, which we'll talk about all these examples later. You can pause the video, go back and answer these 3 questions. Let's get into some of the physiology here. So we talked briefly in the last chapter, chapter 6, about this idea of a membrane potential and how there's some type of electrochemical gradient that is keeping the cell stable. Well, the cytosol and extracellular fluid, so inside the cell and outside the cell, have different compositions. There's slightly more positive stuff outside of the cell. The inside of the cell is slightly negative. So this unequal charge distribution is called a membrane potential. The word potential is referring to the potential energy that this cell has to do something. So again, this results from differing permeability to different ions. There's also an active transport process that we'll talk about. It's called the sodium-potassium pump, all keeping this membrane potential stable. Here you see what I mean. Here's the plasma membrane. Here's the outside of our cell, right? Our phospholipid bilayer. Outside the cell, we're very, very positive. Inside the cell, we're fairly negative. Membrane potential. So let's talk about these ions. In the extracellular fluid, you have very high concentrations of sodium and chloride. Inside the cell, you have lots of potassium and negatively charged proteins. So because of this charge distribution, the cell is slightly negative. Ions can typically only enter the cell through membrane channels. Some potassium and sodium channels will always be open. Some of them respond to neurotransmitters, and some of them will change in response to a membrane potential. So if the membrane potential changes, then they will open or close. They can also get in and out of the cell through something called active transport. The sodium-potassium pump is an excellent example of this. This is where 3 sodium ions are pumped out and 2 potassium ions are pumped in. So here's the sodium-potassium pump in action. Our potassium is constantly leaking out of the cell. Sodium is constantly leaking into the cell. These are called leak channels. They're passive. They're always open. Not always, but most of the time. In a relaxed state, they're always open. So this sodium-potassium pump takes the potassium that's accumulating on the outside of the cell and pumps it in, and takes the sodium that's accumulating on the inside of the cell and pumps it out. So changes in this membrane potential can trigger a muscle contraction, as we saw in the last chapter. It can trigger a gland secretion. It can also transfer information in the nervous system, and that is what we're concerned about in this chapter. So let's talk about the resting membrane potential. The potential of a cell at rest is called the resting membrane potential. Once a stimulus arrives, there's a small change, which is called a graded potential. If the graded potential is large enough, then an action potential is going to be triggered, and this is what's propagated or communicated down the axon to another neuron or to an interneuron or to a motor neuron, etc. So the synaptic activity, this is what's going on at that gap in between neurons, or neurons and interneurons, or neurons and effectors. So the presynaptic cell releases neurotransmitters. This is the same thing we learned in the last chapter, right? That motor neuron released the neurotransmitters, and the postsynaptic cell, which happened to be a muscle cell, responded. So the neurotransmitters bind to receptors on the postsynaptic cell. In the last chapter, we were talking about the postsynaptic cell being a muscle fiber. Here, we're talking about it being probably another neuron. That changes the permeability on that postsynaptic cell. So thousands of axon terminals are communicating with that neuron cell body, stimulating or inhibiting the neuron. It's going to either impact that neuron and make it send an action potential, or it's going to turn that neuron off and have it not send an action potential. So here's where all the magic happens. You have this multipolar neuron receiving information. It's at rest, and then all of a sudden something comes in, a stimulus. It can produce a graded potential. The graded potential could be small. The cell could say, not enough. That stimulus wasn't strong enough. I don't really need to let the other cells know about this. Or it could be a really strong potential, where several several stimuli are coming in saying, we need to respond to this now. So this graded potential kind of tips the scale, and it's like a domino effect, and the action potential propagates down the axon. Then at the synapse, those neurotransmitters are released, bind to the receptors on the postsynaptic cell, and then the postsynaptic cell can respond. So pause the video, go back and answer these questions. Let's talk about the phases of an action potential. Unequal charge distribution is called polarization. So when the cell depolarizes, this is when a neurotransmitter like acetylcholine opens up the sodium channels. Those positive sodium ions are going to rush into the cell, making our cell now less negative. In other words, making it more positive. Repolarization is when the opposite happens. Those sodium channels close, potassium channels now open, and potassium floods the cell. It leaves the cell. It goes out of the cell. And now our membrane potential comes back down and becomes more negative. here's what happens, right? These axons are coming in, communicating to our cell, releasing a neurotransmitter like ACH. Our sodium channels open, this depolarization that you see, and then once it reaches a certain level, those sodium channels close, potassium channels open, potassium leaves the cell, and our cell repolarizes and goes back to normal. So let's talk about how these action potentials are generated. That graded potential has to be there. If depolarization reaches threshold point, again, that tipping of the scale, that graded potential is strong enough that it makes our cell need to communicate to the postsynaptic cell, then the action potential is triggered. So you have neurotransmitter release on the cell to open the sodium channels. That's causing a very large depolarization. Our sodium channels then close, the potassium channels open, and that's going to cause the repolarization. And then the sodium-potassium pump works to get back to normal, resting membrane potential. Signal comes in, causes a graded potential. That's going to cause sodium to rush in, potassium to rush out. And then the sodium-potassium pump gets it all back to normal. Now, because our axons can be myelinated or unmyelinated, this happens in two different ways. It can be a continuous propagation. In other words, the action potential at one location triggers the next location, triggers the next location, triggers the next location, etc. It just keeps depolarizing and repolarizing and sending that signal down the axon. It can take a relatively long period of time. Saltatory propagation is when the myelinated axons insulate or protect part of the axon. So that signal is going to depolarize here, and then it's going to depolarize here. Instead of having to depolarize at every single stop along the way, it's skipping to the nodes. It's skipping to the unmyelinated parts, which is why it's faster. It's a faster way of propagation, a faster way of information sending. So here you see with saltatory propagation, that signal is only having to depolarize at these special locations. Otherwise, it's going to have to do it at every single segment along the axon, taking a much longer time to send the same signal. So propagation speeds are the fastest along very large diameter axons that are myelinated. Urgent information, life-threatening information is carried along these large diameter myelinated axons. Less urgent information can be carried on unmyelinated fibers. This is most of our visceral stuff. So like the fact that our digestive food is now ready to move from the small intestine to the large intestine, not a life or death situation. It'll get done eventually. But something like you're touching a hot stove and you need to remove your hand, absolutely. Something like a loud flash goes off over there and you need to turn to look at it, that's going to happen quickly. So pause the video, go back and review. Let's keep going. Let's talk about the synapse. Action potentials propagate along that axon and eventually arrive at the synapse. This is where the transfer of the action potential from one neuron is going to be transferred to another neuron. The presynaptic cell forms what's called the axon terminal. This is where the synaptic vesicles hold special neurotransmitters. There's a small space and then the postsynaptic cell. That space is called the synaptic cleft. So here you see the anatomy. Our axons coming down, our axon terminals here, synaptic cleft is this little space, and you see lots of vesicles containing special neurotransmitters. So here's the overall process. It's the same thing we learned in the last chapter. Action potential comes down, our vesicles release our acetylcholine into the, is it acetylcholine in this? Yeah, it's acetylcholine. Release that into the synaptic cleft, binds to receptors. Those receptors will then open the sodium ion channels. Sodium can now rush into the cell, depolarizing that and causing that action potential to continue occurring. And acetylcholinesterase, ACHE, is sitting around in that synaptic cleft, waiting to gobble up and recycle the acetylcholine. So there are over 100 different neurotransmitters. We've just been talking about acetylcholine because that's found at the neuromuscular junctions between neurons and muscles, but they all work in different ways and they all have specific functions. Here are some of them. Do you need to know all of them? No. You should be familiar with what a neurotransmitter is, and you need to know just these 5 that are listed here on this page. And these are ones that some of you are probably already familiar with. Acetylcholine, as we said, this is the most studied. This is the one at neuromuscular junctions. Norepinephrine, this is involved in our pituitary gland secretion, overall attention, and consciousness. Epinephrine, this is our excitatory. The norepinephrine is usually kind of an inhibitory neurotransmitter. Epinephrine is the other one. Epinephrine is more excitatory. Serotonin, this is our emotional states. Glutamine is inhibitory. It's blocking the entrance of calcium, so it might be slowing something down like muscle contractions or heart rate. So you can pause this, go back, and answer these 3 questions. Let's talk about the functional anatomy. So we were just talking about the microscopic physiology, the cells, etc. Let's talk big picture now. Let's get into the brain and spinal cord. Our spinal cord and our brain, this is the majority of the nervous tissue in our body. The brain's carrying 97% of the nervous tissue. So it's mostly up there. Weighs about 3 pounds. Males have bigger brains than females because they just have bigger bodies. There's no correlation between brain size and intelligence, so don't think it's better for you guys. All right, the overall development of the central nervous system, it basically starts as a neural tube, and then that tube just kind of bubbles out into little sections and eventually forms. Here you see what I'm talking about. Do you need to know all of this? No, but it's good for background knowledge. The neural tube starts to bubble and form different areas. This forebrain is basically going to blossom and flower over the rest of the brain and develop into what we call the cerebrum. And here you see that process happening. Do you have to know all of this? No, I'm covering it just for background. It is in your book, so I figured I would mention it, but it's not going to be on a test or quiz. Let's talk about that cerebrum, that outer shell of your brain. It has 2 major hemispheres. It's surrounded by, or sometimes called the cerebral cortex. That's the outer layer. This is our conscious thought. This is our memory and storage and processing of information. This is sensory processing. This is regulation of skeletal muscle contractions. On the surface of the brain, there are fissures, there are gyri, and there are sulci. Fissures are really deep grooves, like in between the 2 hemispheres of the brain. There's a deep longitudinal fissure, right? But the brain itself has little mountains and valleys on it. Those are the gyri and sulci. So the cerebellum is the second largest brain structure. That's that little walnut-shaped piece on the back. This is coordinating our body movements and motor commands. Then you have what's called the diencephalon. This is kind of inside the cerebrum. The cerebrum is kind of this outer shell, and the diencephalon is this little popsicle-like structure on the inside, or lollipop-like structure on the inside. This is where your thalamus is. Your thalamus is relaying your sensory information. And then your hypothalamus, which is all of your hormone control. There's also the midbrain, pons, and medulla that make up the brain stem. So the midbrain is visual and auditory information. This also helps maintain consciousness. So this is helping your sleep-wake cycles. Your pons is connected to the cerebellum via the brainstem, and this is our visceral and somatic motor control. And then the medulla oblongata is basically our centers for heart rate and respiration, as well as relaying information up to the thalamus and brainstem. So here you see all of this. This is why I was calling the diencephalon a lollipop. Kind of looks like that. The cerebrum on top, you can see a deep fissure here. And then there's, again, a fissure in between the 2 hemispheres. The gyri are the mounds. The sulci are the little tiny grooves. Here's the cerebellum, that walnut-like structure, the spinal cord, medulla oblongata, pons, and midbrain all up in there. And then the diencephalon, which is the thalamus and hypothalamus. You can't see that from the outside because it's all covered by the cerebrum. So let's talk about the ventricles in the brain. Did you know that your brain has space in it? It's not just a solid ball of neural tissue. So the ventricles are special passageways that are filled with cerebrospinal fluid. They are lined with special cells called ependymal cells. Do you remember what they do? We just talked about them about 10 minutes ago. Ependymal cells, remember, secrete and monitor the cerebrospinal fluid. There's 2 lateral ventricles in the cerebrum. There's an interventricular foramen that connects the lateral ventricles to the 3rd ventricle. The 3rd ventricle is within the thalamus, within the diencephalon. And then the cerebral aqueduct connects that one to the 4th ventricle, which is located right near the pons. Here you see them, our lateral ventricles in our two hemispheres, our interventricular foramen, followed by the third ventricle, and then, yes ma'am, the cerebral aqueduct. I will, I gotta finish this lecture, sweetie. Why are you not getting my food? I did feed her. I just fed her. I'm not starving my child, I promise. Fourth ventricle here kind of looks like a little sideways triangle in front of the pons. And then this continues to the central canal all the way through the spinal cord. So cerebral spinal fluid circulates throughout all of these ventricles and then out around our brain and spinal cord, as we'll see in a little bit. Let's talk about some connections in our brain. The corpus callosum are white tracts of matter that connect our two cerebral hemispheres. There's about 4 billion impulses per second. Wow! Relaying information right to left, right to left, right to left. The nuclei is a certain term. It's not referring to the organelle of a cell. It's referring to a group of cell bodies within the central nervous system. Do you remember what group of cell bodies outside of the central nervous system is called? A ganglia, right? In the central nervous system, we call them nuclei. The basal nuclei is a special group of cell bodies which are involved in coordinating movements like walking. You don't really have to think to walk or think to run. Those basal nuclei are there, and once that pattern is established, they automatically do it. My dog's just sniffing around. Here you see the corpus callosum, the right and left, and some of our ventricles from this anterior frontal section. You can pause the video, go back and review. Let's talk about the meninges. The meninges are the coverings of our brain. You have the outermost layer, which is called the dura mater. It has an inner and outer fibrous layer. There's a small gap in between. These cover the dural sinuses. It's basically collecting the venous blood. It's found in the special little gap. And then there's an outer layer that's fused with the periosteum on the underside of the skull. So typically, if you're doing a dissection or if you're in a head injury and they have to do surgery, when they take your skull off, they take your cranial bones off, the dura mater is automatically going to be fused with the periosteum of your skull bone. Same thing with the arachnoid matter. That outer epithelial layer is supported by a fibrous meshwork, and the subarachnoid space will separate it from the pia mater, which is the innermost lining. The pia mater is practically fused with the surface of the brain. It's very thin and delicate. Its whole function is there to hold the blood vessels in place. So here you see what I mean. The dura mater is thick and fibrous, connected and fused with the periosteum of the skull bone. Then our arachnoid matter, you can see why it's called arachnoid. These little projections look like spider web, and this subarachnoid space is where your cerebral spinal fluid will circulate, and the pia mater sitting right on top of the brain tissue. So there's also dural folds. This is invaginations of those meninges and of the connective tissue. The dural sinuses are the collecting veins within the dural folds. So they're basically just little pools where the blood will sit and wait to be drained out. The falx cerebri is the dural fold between the cerebral hemispheres. There's also the tenatorium cerebelli. That's between those cerebral hemispheres and the cerebellum. And then the falx cerebelli is between the cerebellar hemispheres, because the cerebellum itself, yes, it's a walnut, but it has two hemispheres to it as well. Here you can see these dural folds kind of coming in. They're just the connective tissue that's covering all of the compartments of our brain. Let's talk about this cerebral spinal fluid. It surrounds and bathes the surface of our central nervous system. It's produced by the choroid plexus. The choroid plexus is a specialized area of ependymal cells and capillaries spread throughout. It basically flows from the choroid plexus through the ventricles, through different spaces, eventually ending up in the subarachnoid space around the brain and the central canal of the spinal cord. It's absorbed into the venous blood at locations called arachnoid granulations. About 150 milliliters of CSF is what we have in our body. Here you can trace the pathway. Here you see the choroid plexus kind of lining the area we call the fornix in our diencephalon area. It's made there. It circulates out through the arachnoid granulations out here, or out through the subarachnoid space, out down the ventricles through our central canal, again, out through that subarachnoid space. And then at special locations, as you see here, called arachnoid granulations, it's reabsorbed into the blood. This is just a closer look at that choroid plexus. You have the vessels here and the special choroid plexus ependymal cells exchanging materials to produce cerebrospinal fluid and take the waste out, put it back into the blood. So in other words, our brain tissue does not touch our blood. There's a few exceptions, but for the most part, it doesn't. That's the arachnoid granulation reabsorbing. You can pause here to go back and review. Let's talk about the lobes of the cerebral cortex. Each cerebral hemisphere is divided into regions called lobes. You have a frontal lobe, a parietal lobe, a temporal lobe, an occipital lobe. If you know the bones of the skull, you know the lobes of the brain. There's also a fifth lobe called an insula, which is medial to the lateral sulcus. It's just kind of inside the sides of the brain. We'll look at some pictures. There's a lateral sulcus. These are some superficial landmarks. A lateral sulcus, a central sulcus. There's a precentral gyrus and a postcentral gyrus, and then a parietal occipital sulcus. Remember, a sulcus is a groove, a gyrus is a mound. So let's look at some of these features. Here's the lateral sulcus between the temporal and frontal parietal lobes. Let me explain how I'm going to test you on these things so that you understand. I will be referring to them in context. I will not give you a diagram. You will not have to label or color or identify anything. You may have to do that in lab, so just be aware. The central sulcus is here, dividing the frontal and parietal lobes. It's also dividing our our primary sensory cortex, which is right here on this postcentral gyrus, from our primary motor cortex, which is right here on this precentral gyrus. Here you can see these a little bit better on a sagittal view, a mid-sagittal view. You can see areas of our diencephalon and the different lobes, our central sulcus, pre and postcentral gyrus. It's always important just to know front from back or anterior from posterior, and you can very easily do that by locating the cerebellum, which is always posterior. Let's talk about these cerebral hemispheres. There is crossing over that occurs. Sensory information from one side of the body will eventually end up on the other side of the brain. That's why if you're right-handed, that means the dominant side of your brain is your left side of your brain, and vice versa. Motor commands from one side of the brain will eventually end up on the other side. This crossing over occurs in the brainstem and spinal cord, depending on which pathway. It does have specific important functions, but for the most part, each hemisphere does the exact same thing. You could pause the video and go back and review. Let's talk about some of the specific functions of these cerebral hemispheres or cerebral regions. First, you have association areas. Association areas are areas that are specific to interpreting incoming data or coordinating motor responses. There's a primary motor cortex. This is where special cells called pyramidal cells, they look like pyramids, send voluntary commands to the skeletal muscle system. There's also a somatic motor association area. This is coordinating our learned movements. So this is like you get to a set of stairs and you can walk up them. If you've ever watched a toddler or baby try to learn how to walk upstairs, it's a process. They have to understand how to step forward and step up at the same time. So once you have those skills, you don't have to think about them anymore. There's a primary sensory cortex. This is receiving general somatic sensory information like pain, touch, pressure, vibration, temperature, etc., whether or not the seat that you just sat down on was warm or cold or whatever. There's also a somatic sensory association area for our sensory cortex activity, like recognizing when you're being touched or when the hairs on your arm are moved by a breeze. You have a primary visual cortex receiving visual information from the thalamus, a visual association area, which is where you're interpreting data. So this isn't only that you're looking at something and I'm looking at my keyboard right now, it's black and it has all these boxes on it, but this is looking at it and saying, this is a keyboard, those are letters, those are numbers, this is what that does. It's all done in a visual association area. The gustatory cortex receiving taste receptors, olfactory cortex receiving information from smell, the primary auditory cortex receiving sound information, and then the auditory association area where you take that sound information and you understand what it means. If you've ever listened to a foreign language before, your auditory association area is like, what the heck, right? If you've listened to someone speak a different language, you're hearing it, that's your auditory cortex, but you have no association. So your auditory cortex is not responding. Here they all are on the brain. Again, I'm not going to ask you to find it. You may have to do that in lab. Let's talk about integrative centers. These are more complex processes. These are restricted to either the right or the left side. They're not on both sides of the brain. The speech center or Broca area or motor speech area, this is regulating our pattern of breathing and speaking. This is very active while you're speaking. This is how you know how to move your tongue, and you know when to take breaths, and you know how to move your lips to form words. So it's not just knowing the word, it's actually knowing how to say the word and how to communicate. The prefrontal cortex, this is our predicting, this is our judgment, and this is all abstract intellectual stuff in the prefrontal cortex, very, very front part of the brain. There's also a frontal eye field, which is our learned eye movements, like how to read a text, how to glance at something and glance back up, kind of what I'm doing now. So I'm looking at the PowerPoint and then trying to look at the camera, which is always awkward, right? And just looking at this little black thing, trying to do that so that you see me looking at you as I'm speaking. There's also the general interpretive area. This is usually only on the left hemisphere, and this is integrating sensory information with visual and auditory memories. So you're looking at something, or maybe you're smelling something, you say, oh, that reminds me of my grandmother's kitchen, right? Or you see something, you're like, I've been here before. Oh yeah, I remember this is there and this is there, right? We've all had those times of moments in our life. Here is some of these areas on the brain. Let's talk about lateralization. The left hemisphere, and what lateralization means is basically the small differences between the right and left sides of the brain. So the left cerebral hemisphere, this is interpretive and speech. This is very language-based. This is analytical, math, and logic. So again, if you're right-handed, this is your strong stuff. Right cerebral hemisphere is interpreting sensory information. This is really more of a sensing and emotional side. So if you're left-handed, this side of your brain dominates what you do. Left-handed people tend to be more emotional rather than analytical. So this is just showing you that lateralization, some differences between the two hemispheres. Let's talk about handedness. As I've mentioned, the premotor cortex controls hand movements. It's larger in the left hemisphere for right-handed people. About 9% of the human population is left-handed. So if you're a lefty, congratulations, you're in the minority, as you already know. There's a very high percentage of artists, musicians that are left-handed, again, because of that lateralization. The primary motor cortex and association area in the right hemisphere is near the association areas for spatial, visual, and emotion. So dominates on left-handed people. You can pause the video, go back, and review. Let's talk about moving from the cerebrum into that little lollipop structure on the inside called the diencephalon. This is surrounding the third ventricle where cerebrospinal fluid circulates. It's formed by the thalamus and the hypothalamus. There's also a little gland in there called the pineal gland. Pineal gland secretes melatonin that regulates our sleep-wake cycles. Let's talk about the thalamus. The thalamus is actually two halves. It's separated by that third ventricle. It's a relay point for sensory information. When sensory information comes into the body or into the nervous system, it always goes to the thalamus first, and then the thalamus can send it to wherever it needs to go. Send the information to the primary sensory cortex or maybe even to the subconscious of the brain. It's also very strong in coordinating voluntary and involuntary motor commands. So you're running and you see that there's a log in the middle of the trail, and so your thalamus sees that because that's sensory, right, and interprets that, and then you're going to have to respond by jumping over the log. So all of that happens quickly while you're still running and breathing and, you know, observing and etc. The hypothalamus has lots of centers in it. There's a subconscious center with some emotions like rage and pleasure and pain. There's also a adjustment center in there for adjusting and regulating the pons and the medulla oblongata. Pons and medulla oblongata, as we'll see, is our respiratory and heart rate. This is also coordinating nervous system and endocrine hormone system activities. There's also several hormones that come out of the hypothalamus, ADH and oxytocin. Hunger and thirst are regulated through a hypothalamus, again, associated with emotions, which is why there's emotional eating and stress eating and all of that. It's all, you can thank your hypothalamus for that. It's also coordinating voluntary autonomic functions like the digestion of your food. Regulating body temperature is all done in your hypothalamus. It's your body's thermostat. And coordinating your daily activity cycles like sleeping, waking, napping, etc., all done through the hypothalamus because the pineal gland is off of the hypothalamus and the pineal gland secretes that melatonin, as I mentioned. There it is. Let's talk about the midbrain. The midbrain has two pairs of nuclei. Remember, nuclei is just a cluster of cell bodies. They're called colliculi. You have a superior colliculus and an inferior colliculus. The superior colliculus is controlling reflexes to visual stimuli. Boom, I'm coming at you. Oh, you're going to blink, right? That's the superior colliculus working. Inferior colliculi is auditory stimuli. You hear something over there, and you look at it all of a sudden. That is your inferior colliculi working. It also contains motor nuclei for ocular motor, trochleocranial nerves. We'll learn about cranial nerves in a little bit. The reticular formation, which is involved in sleep-wake cycle and emotions. It also is there to help control our muscle tone, our posture. It's also regulating the basal ganglia through something called the substantia nigra. This is just a cluster of kind of darkened cells, cell bodies that, again, coordinate movements. The cerebral peduncles are linking parts of the cerebrum and cerebellum to the brainstem. So they all come through the midbrain area. So you can see, where's my mouse? Here's our diencephalon, and now we're getting into the brainstem. Here's our midbrain. Then we're going to go to the pons, and then the medulla oblongata and the cerebellum. Here's a superior and inferior colliculus, substantia nigra, just a relay center there, and all of the different fibers connecting our midbrain to the pons. So midbrain is processing auditory and visual information and generating a reflex. The pons is sensory information to the cerebellum and thalamus, and this is also subconscious, somatic, and motor and visceral motor control. So again, stuff that you don't have to think about: our respiration, our digestion, our heart rate, and the medulla oblongata, which is basically connecting the brain to the spinal cord. And it has lots of cranial nerves rooted from the medulla oblongata because our cranial nerves control some of our basic functions. It's also our cardiovascular centers: heart rate, blood pressure, strength of contraction, all controlled through the medulla oblongata, and our respiratory rhythmicity centers setting the pace for our breathing. There it all is. Let's talk about the cerebellum. This is our automatic processing center, looking at our body position. When you fall and trip and you reach out your arm to catch yourself, you can thank your cerebellum for that. When you're sitting on a bench and you're, keeping your body centered so you don't fall off, that's all done through your cerebellum. You don't have to consciously think about those things. Your body automatically adjusts for you. It's composed of mostly white matter and some neural cortex called the cerebellar cortex. There you see it. Oh no, it looks blue there. It's not really blue. You can pause the video, go back and review some of the information. We're winding things down with some specific parts of our central nervous system here, looking at the reticular formation, which is generating involuntary functions. It contains something called the reticular activating system. The reticular activating system kind of does exactly what you think it does. It's activating your upper consciousness, making you aware, making you awake, right? You may be sitting here listening to me go on and on and on, and you might be dozing off. That means your reticular activating system is shutting down. Then all of a sudden I say, wait, this is important. This is going to be on the test. And now your reticular activating system says, whoa, we got to wake up. We got to pay attention. All right, what is she going to say? That's your reticular activating system coming up and about and making you more conscious and more aware. So it's taking input from visual as well as auditory and generating this overall conscious response in the cerebrum. You can pause the video, go back and review. An electroencephalogram, or EEG, is measuring your brain waves. Billions of neurons producing a chemical electrical impulse is going to be able to produce enough energy to be measured and detected. And that's exactly what an EEG does. You put some electrodes on or around your head, and that electrical activity can be measured. The EEG can be used to diagnose abnormal brain waves or abnormal brain activities, such as seizures and even seizure disorders like epilepsy. All of our brains emit waves at all times, unless we're dead. Alpha waves are normal, healthy, awake brain waves. You can also be resting with your eyes closed and alpha waves will still be there. Beta waves are usually emitted when you're under stress. You're nervous, you got to study, you got all this stuff to do, oh my gosh, and this is that franticness that goes on in your brain, and those can be detected as beta waves. They're slightly different than alpha waves. Then you have theta waves. Theta waves are seen in children. They're also seen a little bit in sleeping adults, maybe adults that are so frustrated they can't even verbalize what they want. In some cases, they could also show a brain disorder. Children, and if you think about sleeping adults or adults that are frustrated or children, have a limited vocabulary during those times, right? Children don't have a huge range of vocabulary. They might not know what something is, right? Same thing with frustrated adults. You're like, whatever your name is, get over here, right? And that's because those theta waves have taken over and you're not allowed to, you are incapable of verbalizing exactly how you're feeling. I don't know, I'm just mad, right? Those are your theta waves taking over. Delta waves are sleep. I always think of delta like sleeping on a plane, like Delta Airlines. That's what I always think of. So this is sleeping at all ages. Infant brains, awake adults with brain damage can show delta waves as well. Here's what they look like. Do you have to know what they look like for me? No, maybe for lab. You can pause the video, go back and review EEG stuff. Let's talk about the spinal cord. Last few pages of the chapter is dedicated to the spinal cord. It's about 45 centimeters long, or about 18 inches. It has an outer layer of white and inner layer of gray, which is opposite of the brain, right? The brain had the gray matter on the outside and the white matter on the inside. There's a cervical enlargement, which is in the neck region. This is where all the cell bodies come off to supply the neck, arms, shoulders. There's a lumbar enlargement, which is going out to the pelvis and lower limbs. The conus medullaris is the tapered posterior, kind of inferior section of our spinal cord where everything kind of dissipates to, and it's forms something called the cauda equina, which looks like a horse's tail. That's what cauda equina translates to in Latin. Here you see it, the cauda equina. The conus medullaris is this kind of end point right around L1, L2, cervical enlargement up here, lumbar enlargement up here just above the lumbar vertebrae. So there are 31 segments which coordinate to our associated vertebrae. There's a posterior median sulcus and an anterior median fissure, just a little deeper. And there are spinal nerves that come off of our spinal cord. There's a dorsal root, which is where all of the axons of the sensory neurons are found, and a dorsal root ganglion, which is holding all of those cell bodies. The ventral root holds our axons of our motor neurons. Here you see it a little better. The white matter, this is the anterior median fissure, posterior median sulcus. The gray matter is on the inside. On the back is the dorsal root, which is all of our sensory information. Ventral root is on the front side, and then the spinal nerve is the fusion of the two. So you can pause the video, go back and review. Let's talk about gray matter organization of the spinal cord. The gray matter in the spinal cord forms like a little butterfly shape in the middle. They're called horns. There's a posterior gray horn, which has the somatic and visceral sensory nuclei. The lateral gray horn, which has visceral motor nuclei, usually only associated with the abdominal area. And then the anterior gray horn with the somatic motor nuclei. The cell bodies of the neurons are called nuclei, again, because we're in the central nervous system. You have the sensory nuclei. These are receiving and relaying information from the peripheral nervous system up into the brain, and then the motor nuclei, which are issuing commands out to the peripheral nervous system. Here you see an actual cross-section or a cartoon version. I always like the cartoon version. They're just a little bit more easier to define. There's the gray matter on the inside. The horns are just the separate areas, right? Posterior, lateral, anterior. All right, now we get to the white matter. The white matter is called columns. Gray matter's horns, white matter's columns in the spinal cord. You have a posterior white column, a lateral white column, and an anterior white column. The columns are containing tracts or bundles of axons, which are coming and going. You have ascending tracts going up to the central nervous system and descending tracts carrying that motor commands out of the central nervous system to the periphery. So here you see the organization of the gray matter nuclei and our white columns. You can pause the video, go back and review. We are winding things down. Let's talk about spinal meninges. Same thing as cranial meninges. Only big difference is here we're talking about surrounding our spinal cord instead of our brain, but it's the same thing. Dura matter is tough, fibrous, and we have an epidural space here between the dural matter and the vertebral canal. And that space has lots of blood vessels and fatty insulating adipose tissue. But otherwise, it's the same exact stuff as the cranial meninges. Here you see it. You can see that epidural space is just between the dura mater and the vertebrae. Very, very small space. The spinal tap is where they're taking they're actually puncturing in through the skin, in through a little gap of, it's usually between L1 or L2 in your lower back, which there's a little bit more of a significant space there. It's just under the lumbar enlargement. And they're able to extract some cerebrospinal fluid, and they can test your cerebrospinal fluid for bacteria or whatever. They can also administer anesthesia in that area. So again, that epidural space, typically they put it just below L1. Here are the review questions for this section, and that concludes chapter 7. So hopefully I didn't talk too fast. I know I did. That's the joy of having it all recorded. You can go back and re-listen to it. And make sure you read your textbook or check out the Mastering A&P website for fun, interactive activities. All right, guys. See you later.